I'm going to cut out most of the immune system, and that's commonly what I do in the class because the immune system is something that you really can't cover in one lecture. I'm going to talk about a couple very basic things about the immune system. Then I'm going to talk about cancer, and then we're going to turn our attention to carbohydrates. So that's the plan for the day. Last time I finished talking up about gene therapy. And what I do want to say about the immune system really relates to what it does and how it functions. Okay? So the immune system is a multicellular organism's protection against the rest of the world. And that protection against the rest of the world manifests itself in the form of recognition and binding. And that recognition and binding happens as a result of production of special proteins called antibodies. All right? So antibodies are proteins produced by the immune system, and they're produced in response to something that the body perceives as an invader. So they're produced in response to something that the body perceives as an invader. Okay? They are proteins. And the immune system itself has an incredible complexity, as I said, way more than I'm going to talk about in one lecture here. But suffice it to say that our body is capable of producing 10 to the 12th different antibodies. Antibodies, again, being the proteins that do the work of the immune system. 10 to the 12th. That's 10 to the 12th different proteins that the immune system can produce. Okay? Now, if you do your math, and you know that the human genome has 7 billion base pairs to it, you recognize that 10 to the 12th is a trillion, and that a trillion is considerably bigger than 7 billion. How is it that the immune system can make way more proteins than there are base pairs in the human genome? That seems a little odd. Well, it turns out that the way the immune system does it is by shuffling the deck, as it were, mixing and matching segments. And so even though we may have a deck of cards that only has 52 cards, when we shuffle that deck, the number of combinations that we can get for the way the cards rearrange themselves is incredibly enormous, right? So you would be hard-pressed to ever in your lifetime shuffle a deck of cards and have it come up twice the same exact order of cards. So that's how the immune system actually gets the diversity that it does. And again, that's, as I said, beyond the scope of this course. The antibodies are the workhorses of the immune system. And the antibodies have a structure that looks something like what you see on the screen here. That it consists of basically 4 chains. And the 4 chains are 2 chains that we refer to as heavy. And you can see them on the outside of this Y-shaped structure here. And 2 chains that are referred to as light, and of course they're smaller, and you see them on the inside of the Y-shaped figure on the screen. You also see connections between the chains, and those connections are typically disulfide bonds that help to hold everything together. Okay? The chains have within them regions that we talk about as being constant and variable. Okay? So they have regions that are constant and regions that are variable. The constant regions of the chains are largely those at either this end, right here, or this end with respect to the light chains. All right? So the further we go up, the more variable we get with respect to these chains. The constant is not absolutely constant, I should point out, but they're fairly constant from one antibody to another. There are several major types of antibodies, and all antibodies of a given type will have the same constant regions. We don't need to go into that. Okay? The important thing for our purposes is up here. It's the variable regions, because it's the variable regions that recognize and bind to what the body perceives as foreign. And that binding is very specific for the structure of that foreign object. That foreign object could be a protein. That foreign object could be a polysaccharide. That foreign object could be a nucleic acid. But if the body perceives it as foreign, the antibody will attack it. And it attacks it and binds to it. All right? Now, the variable region for any given antibody, once we have an antibody that recognizes an invader, for example. So let's imagine that if you are out running around and you get exposed to a virus. This could be by either by infection or by injection with a vaccine. Okay? Either way, we accomplish the same purpose. The thing that's in your body is something that's not made by your body. It's not a protein made by your body. It's coming from a virus, and the body says, that's not me. Okay? The immune system is also important in rejecting organs. So if you get a heart transplant, your immune system is the part of your body that's recognizing, this isn't my heart. Okay? And it will attack it. That's what immune rejection is all about. Okay? Now, the key is that this variable region recognizes and binds to things. And so if you have an invader that comes in, let's say you have a protein from a cold virus that's there, and the body says, this isn't me, the antibody that binds to that is a specific antibody. Its region will be different than the variable region of other antibodies. But once one of these guys has bound to that antigen, it will be stimulated to be produced more. So the body will start making millions and billions and zillions of this one that recognizes and binds to that foreign invader. Okay? So that's how the immune response actually manifests itself. That's why vaccines work. You get a vaccine because the body recognizes what you've introduced into the body as a foreign invader. The immune system attacks it. The immune system makes antibodies against it. And if you get infected again by that same thing within a span of about 10 years, the immune system has a memory that it will go back and remake this antibody that it recognized before. Okay? So it's a very cool system. I don't know where people stand on vaccines, but it's incredible to me to think that we live in a world where we have vaccines available and there are people who think that the vaccines are evil in some way or dangerous in some way. Okay? It's one of the worst possible things that we could have. We have measles developing in the richest country on the face of the earth because people think that vaccines are problems. Okay? That's a very scary thought. And the reason that happens is because there's not enough people who get vaccinated. All right? There are the likelihood of dying from not getting vaccinated compared to getting vaccinated is millions of times greater for not getting vaccinated than it is for getting vaccinated. And it's important to get vaccinated as a civic duty. Okay, that's kind of heavy language, but as a civic duty to protect those who can't be vaccinated. People with a compromised immune system can't be vaccinated. Infants can't be vaccinated. There are horror stories of infants in California dying of whooping cough because their idiot grown-ups won't get vaccinated and stop the spread of this thing. And the infants can't be vaccinated for it. Their immune system has got to get to a certain age before they can be vaccinated. And as a consequence, whooping cough is out there because these boneheads aren't getting vaccinated. Get vaccinated. That's my bottom line to you. Okay. All right. The thing that the immune system recognizes is what we call an antigen. All right? And the antigen is the foreign thing. And you can see here that there are antibodies that are attacking this foreign thing. And this foreign thing is usually a protein, but it's not always. It can be other things besides a protein, but it's usually a protein. This protein might be part of the coat of a virus. It might be a protein that's produced in response to the virus, or it's part of the viral life cycle, or something like that. But if you look at this carefully, you'll think, well, that's interesting. This immune thing that's here, this antigen thing that's here, different parts of it are being recognized by different antibodies. And so when your body fights something with an antibody, it doesn't fight it with just one antibody. One antibody might recognize a specific part of a protein, and there might be millions or billions of those antibodies produced, But another antibody might recognize a different part of the same protein and have millions or billions of those produced. So your immune response is not what we call clonal. Clonal meaning all one type. These are all one type, but these are a different type. And these are yet a different type. So for a given antigen, we might have 50 different types of antibodies that are recognizing and binding to that antigen. Okay? It's what we call polyclonal. Polyclonal meaning multiple versions of which there are millions of copies of each. All right? This is in contrast to something that's called monoclonal antibodies. Okay? What if we could take, let's say, just this specific antibody at the very bottom that recognizes only this one very specific part of this antigen, and we could produce only this antibody? If we could do that, we would produce something called a monoclonal antibody. It wouldn't have all the other ones that recognize other parts of the same antigen. But this monoclonal antibody would be recognizing a very specific part of a structure of one specific antigen. It will be recognizing only this piece right here. This piece right here might be the same as this piece right here. And we would have millions of copies of that one antibody. So when we have that, we have what's called a monoclonal antibody. We don't have things recognizing other parts of the same antigen. Okay? Now, to put a name on top of those things, we talk about the part that's bound by an antibody on an antigen, that is a subpart of the antigen. We call that part, the specific part that's recognized and bound by the antibody, an epitope. So the epitope of this antigen is right there. And this epitope right here is not the same as this epitope right here, or this epitope right here, or this epitope right here. But this is a specific epitope. So an epitope is a specific structure on an antigen that's recognized by an antibody. So to sort of say it in another way, a given antigen might have 100 different epitopes, which 100 different antibodies could bind to. And that would be a polyclonal mix of antibodies. But if I had one antibody that recognized one of those epitopes and only that one antibody, I might have millions of copies of it, but if they're all recognizing exactly the same epitope, I would have a monoclonal antibody. Clear as mud? Yes? Okay, so antibodies are proteins, all right? So, but they make 10 to the 12th different antibodies. That's the remarkable thing. Now, I'm not talking about 10 to the 12th, copies of one protein. I'm talking about 10 to the 12th different proteins. That's remarkable. That's really remarkable. Yes?
So when you were talking about how the antibodies are able to signal for more than is produced when they're is there a precursor to that to say which antibodies to send in, or is there just like a pool of the antibodies already there?
Yeah. Okay, so good question. Yeah, if I understand your question, Derry, you're saying, does the body just send in one antibody and hope it works, or does it send in a whole bunch of stuff and then see what works? Is that the sort of summarizing your question? Yeah. So it's very interesting how the immune system works. The answer is it's the latter. That is, it sends in a variety of different antibodies, and that which sticks gets duplicated. And it's a complicated process. If you're curious, I'd be happy to tell you more. I'm not going to go through it here. But the ones that stick get reproduced. And the ones that don't stick don't get reproduced. And the body is constantly shuffling that deck, as it were. Shuffling that deck so that it's producing different ones all the time. And oh, here's one that's working, and I'm going to start making this one. It's pretty cool. Yep. Other questions? Okay. Well, that's what I want to say about the immune system and antibodies. I want to say just a couple words about cancer and give you some terminology that's there, and we'll talk a little bit more about that later as well. And cancer, we all have an idea about what cancer is. Cancer is basically a cell that is growing uncontrollably. Our cells normally have a cellular cycle that they go through that is regulated. And you've seen a little bit of that regulation already. I talked about P53, for example. That was the quality control protein that recognized if replication hadn't proceeded properly, it would stop the cell cycle in its tracks before division occurred so that everything could be fixed, or the cell could commit suicide. All right? That's really important, and it's really important if you think about it in a multicellular organism, because it's really important that my two legs be at least reasonably the same length. So I want the growth to be controlled so that they're working out the same. My arms being the same, my eyes being the same. Okay? What gives me my appearance is regulated growth. Unregulated growth is going to kill me because unregulated growth is going to take resources away from my other cells that need nutrients, that need connections, that need whatever that is that the other cells need. Unregulated growth is going to kill me. And that's what cancer is all about. So a cancerous cell has lost the controls that non-cancerous cells have within them. It's lost the controls. How are those controls manifest? Well, it's quite complicated how they're manifest, but one of the ways in which the cells are controlled in our body is through hormones. Other ways that cells in our body are controlled are through the control of gene expression. And we'll talk a little bit about that in a bit. Okay? Well, it turns out that people studying cancer back in the 70s were curious to know, is there a genetic basis to cancer? That is, can I find things in the genome that, for example, might cause cancer? And to the surprise of many people, in fact, they found such things. And not only did they find such things, but they had actually known of such things going back to the early 1900s. Okay? There was evidence for there being genes that caused cancer being produced as early as 1900. This was found in a chicken. It was a chicken that they found had cancer, and they found that they could take this chicken that had cancer, and they could take something from that chicken and give it to another chicken, and the other chicken got cancer. They found that that was a contagious form of cancer. Contagious forms of cancer are not common. But because it was contagious, they knew that something had to be transmitted, and that something that was transmitted had to be a gene. Okay? You got an astonished look on your face like, don't tell me I'm... What contagious forms of cancer are there? Well, okay. So they're linked to retroviruses. You've learned about retroviruses. In the case of the chicken, what had happened... let me come back to your question. It's a very good question, okay? What contagious forms of cancer are there? If I know somebody who has cancer, does that mean if I hang around I'm going to get cancer? No, it doesn't, okay? Fortunately, cancers in humans are... contagious cancers in humans are very, very rare and very, very not something to worry about. Okay? In cats, for example, they can have a contagious form of cancer, feline leukemia virus. Okay? Chickens, it's called Rouse sarcoma virus and so forth. Anyway, but I'll tell you how that happens in a minute. All right? So anyway, they knew that this was being transmitted in some way, and they knew it had to be a gene. So the term arose for a name for what these things were that caused the cancer, and the name that they settled on was an oncogene. So an oncogene was originally defined as a gene that causes cancer. Well, you might look at a virus and say, hey, here's this virus causes the common cold, the virus causes pneumonia, this virus causes polio. It's not too surprising that virus might cause other diseases like cancer. But it wasn't until the 1970s when people started examining what those genes were that they discovered to their shock that those genes weren't just unique to the virus, but they were found in our genome. We have those genes. Okay? You have in your body several hundred genes that are capable of giving you cancer. Several hundred genes that are capable of giving you cancer. Why do we carry around genes that can give us cancer? Doesn't that seem like it's kind of a dumb thing to do? Well, it turns out that the reason that we have those genes is not so they can give us cancer. The reason we have those genes is because those genes play critical roles in the cellular division process. They play critical roles in the cellular division process. If cells are regulated to divide or not to divide, that regulation has to be at the level of genes. So what if I screw up the genes that do the regulation? Will I get uncontrolled growth? Yes. What if I screw up the way that the cell receives signals from the outside that tells it to grow? Will I screw up controls? Yes. There are many, many ways in which those genes that help control the division process can be screwed up and now result in uncontrolled growth. Well, fortunately, those genes in our bodies don't play that role. They play that role of controlling things. Cells are control freaks, right? When are they a problem? They're a problem when they lose their function. And how do they lose their function? They lose their function by mutation. So when we realized that this was the case, we had to step back and say, well, these oncogenes that seem like they were invaders from Mars are actually invaders from us, and that they're only problems when they mutate. So we had to come up with a new name. We described the oncogenes that were in the unmutated form that are present in every one of our cells every day. The unmutated genes we called Proto-Oncogenes. Proto, P-R-O-T-O, and then dash oncogenes. A proto-oncogene is an unmutated gene that can cause cancer. A proto-oncogene is an unmutated gene that can cause cancer. You mutate that proto-oncogene, your likelihood of getting cancer goes way up. And that's why we worry about mutation. Okay? Well, we've already talked about one proto-oncogene. The proto-oncogene that we've talked about is P53. And I told you that if P53 gets mutated, you're much more likely for that cell to become cancerous because P53 has the ability to put the brakes on when it sees problems in replication. If you don't fix problems in replication, what's going to happen? More mutation, and you start mutating proto-oncogenes, what's going to happen? Cancer. Okay? So there are many proto-oncogenes that are out there, P53 being one of those. And now I'll give you something scary. If you like to go to the campfire and sit around and tell horror stories, right? Okay. When I go to campfires and I tell horror stories, there are different kinds of horror stories. So here's a horror story for you, okay? You can tell this the next time you're at a campfire, and then they'll have you labeled as a nerd for sure, right? Okay. Here's a horror story, okay? We have several oncogenes in our body that if we mutate one specific base pair to any base pair, okay, the likelihood that that cell will become cancerous goes up astronomically. In fact, in cell culture, we can take that one gene and mutate one base pair, and in cell culture, that cell will be cancerous, period. What's the likelihood of that one base out of an entire genome mutating? Well, you've got 7 billion base pairs, one base, one chance in 7 billion. Those are pretty good odds, right? Until you realize you've got several trillion cells in your body. Meaning, it's simply a matter of time until that one base pair goes wonky. You guys glad you came to class today? Okay. That one base pair goes wonky. Now your likelihood of, and by the way, developing cancer is more than one step. I should point this out. In a multicellular organism, you have protections besides fidelity of replication. You have protections of an immune system, for example, which recognizes things. So I'm painting a scarier picture for you than really actually exists. But I paint that picture for you because I want you to think about the importance of maintaining the integrity of your genome. And the integrity of your genome is maintained by avoiding mutagens. Mutagens are things that favor mutation. And we've talked about some of them already. Smoking cigarettes, one of the most common ways to expose yourself to mutagens. Bad water, bad food, and tanning booths. There's 4, okay? The more care you take with respect to all of those, the better off you're likely to be in the long term. Okay, questions about that before I turn to carbohydrates? Everybody scared to death now? Okay. All right, let's talk about carbohydrates. Carbohydrates are something we all know what they are. We have an idea what they are. And I'm going to go through this fairly quickly. So you say, what's new, right? I'm going to go through this fairly quickly because a lot of this you've covered in organic chemistry. One of the things with carbohydrates that you should know is a lot of nomenclature. So I'm going to start with the nomenclature. Carbohydrates are what we commonly refer to as sugars. And those sugars can be combined together in a variety of ways to make larger molecules. They can be complex carbohydrates. They can be simple carbohydrates. Those sugars also are called saccharides. We can have monosaccharides, meaning simple sugar. We can have a disaccharide, meaning 2 sugars joined together. We can have a trisaccharide. You can probably figure out what that's all about. And you can imagine what a polysaccharide is. We're going to talk about all those. The nomenclature that we give for carbohydrates is sugars always are given the name that ends in OSE. OSE at the end of a name means we're talking about a sugar. So we talk about glucose. We talk about sucrose. We talk about galactose. Okay? Those are all sugars, and those sugars have OSE on the end. Sugars, to go back to the nomenclature business, can have multiple carbons. All right? So we can have a sugar that has 2 carbons, be called a diose. 3 carbons would be called a triose. 4 carbons, a tetrose. 5 carbons, a pentose. 6 carbons, a hexose. These are common prefixes. 7 carbons, a heptose. That's not very common, but there are a few. And 8 carbons, an octose. We don't usually see carbons larger than about 8 carbons. I mean, we don't see sugars larger than about 8 carbons. So an octose. Now, those are general names. All right? Among the trioses, for example, I can point to 3 trioses. Okay? And here they are. They occur in a couple of different chemical forms. All right? So sugars can be what we call aldoses, or they can be what we call ketoses. So a sugar that has an aldehyde at its end, and this is one guy that has an aldehyde at its end, is called, this one's called glyceraldehyde. And it's a triose, and it's an aldose because it has an aldehyde at its end. Okay? Another form of sugars that we see are what will be referred to as ketoses. And instead of having an aldehyde at their end, they'll have a ketone at carbon number 2. That's what a ketose will have. All right? So in the case of this guy here, which is dihydroxyacetone, we see that it has 3 carbons and it has a ketone at carbon number 2. One of the things about simple sugars that's interesting is it actually gives rise to the name carbohydrate. The formula for sugars is C with an X below it, where X is the number of carbons. Parentheses H2O, that's water, with an X below it. So for example, if I'm talking about these guys right here, the number of carbons is 3, so I would have C3. I would have H2O times 3, so that means I have C3H6O3. So carbohydrates are hydrates of carbon. That's where the name comes from. They're hydrates of carbon. A tetrose would have C4H8O4. A pentose would have C5H10O5. A hexose would have C6H12O6. Okay? Hexoses and pentoses are the most common sugars that we find. Hexoses and pentoses. Well, before I go and talk about those, I need to remind you of something about your organic chemistry. And that is that if we talked about with respect to amino acids, and that is if we talk about carbons that have 4 different things attached to them, then those 4 different things can be arranged in 3-dimensional space in 2 different ways. With respect to the amino acids, we talked about the L amino acids being the most common, and the D's being extraordinarily rare. We don't see them produced in normal cells. Okay? In the case of the sugars, we pretty much see the D forms. There are some L sugars that we see, right? But we pretty much see sugars in the D configuration. And I'll show you a real simple trick for how to determine that in a bit. So we look at glyceraldehyde. We discover that glyceraldehyde, right here, can have its its structure, can have its 4 things arranged around its asymmetric carbon in 2 different ways. You see them right here. You see D-glyceraldehyde above, and you see L-glyceraldehyde below. The difference between the D and the L? Well, if you see right here this OH, the OH right above the bottom one on the right means it's D, and the OH right above the bottom one on the left means it's an L. That's a simple rule. There's actually a more complex rule that you'll need to know that I'll show you in a minute, but that's a simple way of starting. So if I have a sugar that has an OH on the next to the bottom carbon on the left, that sugar is an L. And if I have a sugar that has an OH on the next to the bottom carbon on the right, it's a D. Biochemists are pretty simple people. Okay. Now, here shows some common sugars that we see. Here on the top is glucose. And looking at it, I would hope that you would recognize that glucose is an aldose. It has an aldehyde group at top. Okay? On the other hand, fructose is a ketose. And you see it has a ketone at carbon number 2. Except for those 2 carbons, the configurations of the OHs on all the other carbons are identical. Notice that carbon number 3 has 4 different things attached to it. Carbon number 4 has 4 different things attached to it. Carbon number 5 has 4 different things attached to it. So each of those is an asymmetric carbon. If I was a chemist, I would go crazy with naming left and right and up and down and R and S and blah, blah. We're not going to do that. That's right. You like that, right? We're not going to. We're simple people. Biochemists are very simple people, right? Contrary to what you might think about me. But anyway, so notice that this is a D sugar because there's that bottom OH, and it's on the right side. This is a D sugar because there's that bottom OH on the right side. But if I were to say on an exam to you, for example, what's the mirror image of D-glucose? And by the way, you will need to memorize the structure of glucose, fructose, and ribose. And that's true both in the straight chain form, like you see on the screen, and also in the ring forms that I'll show you in just a little bit. Glucose, fructose, and ribose. Okay? Now, I don't make you memorize those structures to be mean to you, but rather because I know you're going to need to know them in other courses. Trust me, you will. Okay? So I figure if you learn them here, the next time you learn them, you'll memorize them a lot easier. So that's true in the ring structure and also in the straight chain. The straight chain form, as shown here, is what we call the Fischer structure. The ring configuration that I'll show you in just a minute is called the Haworth, Haworth. Now, getting back to what I was saying about glucose, you'll see here that you guys know what mirror images are, because when we can arrange things in certain ways, they can be reflections of each other. These guys are not mirror images of each other because, for example, A, that's a ketone and that's that's an aldehyde, so that would rule it out. But it's also not a mirror image with respect to the other things. We have left, left, we have right, right, we have right, right. Those have the same configuration. That's not mirror imaged. However, if I were to ask you what the structure of L-glucose was, I would hope that you would remember that L-glucose is not just the bottom OH moved to the left, but in fact L-glucose is the mirror image of D-glucose. I've got to flip every carbon. All right? So here's the OH on the right for D-glucose, OH on the left for L-glucose. OH on the left for D-glucose, OH on the right for L-glucose. Right, left, right, left. So the L form of any sugar is the mirror image of the D form of that same sugar. Okay? The L form of any sugar is the mirror image of the D form of that same sugar. We give a special name to sugars that are to this property of mirror image. L-glucose and D-glucose are what we refer to as enantiomers. E-N-A-N-T-I-O-M-E-R-S. And yes, somebody's going to say, could you repeat that? E-N-A-N-T-I-O-M-E-R-S. Enantiomers. So enantiomers are mirror images of each other. Okay? Enantiomers are a special form of what we call stereoisomers. Stereoisomers are molecules that simply differ in their stereo configuration, the left-rightness, as it were, of the OH groups. If they're perfect mirror images, they're called enantiomers. If they are not perfect mirror images, we call them diastereomers. Yes, I'll spell it. D-I-A-S-T-E-R-E-O-M-E-R-S. Diastereomers are stereoisomers that are not mirror images of each other. Well, let's give an example. Here is L-erythrose, and here's D-erythrose and L-erythrose. Right? From what I've already told you, you should know that the D and L forms of the same sugar will always be enantiomers. And if you look, they are. Here's the OH on the right, here's the OH on the right, here's the OH on the left, and here's the OH on the left. D and L-erythrose are enantiomers. Okay? D and L-threose are also enantiomers. Here's left-right, here's right-left. However, L-erythrose and D-threose are diastereomers. They're not mirror images of each other, but the configuration of their OHs is different. Okay? So these guys are diastereomers. D-erythrose and D-threose are also diastereomers. All right? I'm going to give you one more name. There's a lot of names with this. Okay? Two sugars that differ only in the configuration of one OH called epimers. E-P-I-M-E-R-S. L-erythrose and D-threose are epimers, because the only place that they differ in configuration is here on carbon number 3. They are epimers. So epimers differ only in the configuration of one OH on a carbon. Okay? There's a bunch of sugars. You can see at the top that, and in fact, this is showing only the D forms of the individual sugars. If we were to fully represent everything here, we would double the number of each of these because we'd have to consider the L forms as well. So we would have a D-glyceraldehyde, we would have an L-glyceraldehyde. I'm not going to ask you to draw this. I'm not going to ask you to memorize all these names. Okay? But I just show you this to show you that as we add more carbons, we add more asymmetric centers. And as we add more asymmetric centers, we have many different stereoisomers that can be created. By the time we get to 6 carbons, which is down here on the bottom, the hexoses, the aldohexoses, and these are aldoses because, again, we see that they all have aldehyde at the top. The aldohexoses, there are 16 possible aldohexoses. No, you don't need to know that number. But you can see that every time we add another asymmetric carbon, we double the number. Okay? All right. There's ribose. That's one of the structures you need to know. Right there, on the right. I'm sorry, on the left. Okay? There's glucose. One of the structures you need to know. Okay? Some other common sugars that are on there that I'm not making you memorize include galactose, okay, and mannose. If you looked at glucose and you looked at mannose carefully, you would see that mannose is an epimer of glucose because it only differs in the configuration of this carbon right here. Everything else is the same. Okay. Well, one of the things you learn in organic chemistry is that straight-chain sugars in solution can bend around and form rings. And in solution, one of the most common forms we see is actually in that ring form. We can see the ring happening here. Okay? And that ring can form in a couple of ways. It really depends on the sort of stereoisomeric configuration of the overall molecule. Here's D-glucose in the straight chain form. Okay? D-glucose in the straight chain form. And if we look at this carefully, we see that carbon number 1 can be arranged so that it's very close to the hydroxyl of carbon number 5. And that's what's happening right here. They're just swinging around, touching each other. Well, when that happens, this ring can close, and when it closes, it can close and actually make 2 different forms of glucose. Okay? How do I get 2 different forms? Well, carbon number 1 was not originally asymmetric. It was an aldehyde. It only had 3 things attached to it: oxygen, a hydrogen, and the rest of the chain. However, when it cyclized, that aldehyde became an alcohol, and now that carbon has 1, 2, 3, 4 things attached to it. So the cyclization of the sugar created a new asymmetric carbon that didn't exist before. We've created what we refer to as an anomeric carbon. So the carbon that was the carbonyl group is the carbon that makes the ring. Now, you'll probably blow off what I just told you, but that's an important concept. The carbon that has the carbonyl group is the carbon that makes the ring. In the case of glucose, the carbonyl carbon is the aldehyde. In the case of fructose, the carbonyl carbon is the ketone. All right? Now, I said that that anomeric carbon, which of course was originally the carbonyl carbon, that anomeric carbon now is asymmetric. And if it's asymmetric, it means it can exist in two different forms. And the two different forms we refer to as alpha, if we draw it in the down configuration as shown above, or beta if the hydroxyl is up, as shown below. Now, in solution, this is going back and forth, and back and forth, and back and forth, meaning it's swapping between alpha, beta, beta, alpha, beta, beta, alpha, all the time. It's going back and forth between those different forms. One last term I'm going to give you here, and that is the shape of that ring. Notice that we've got a six-membered ring, and that six-membered ring has an oxygen in it. It's very important in understanding the structure of sugars to count the carbons. Carbon number 1 is where my pointer is. Carbon number 2, in fact, I'll show up for the people on the TV. All right, there's carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, and don't forget we've got carbon number 6 up here. Okay? Carbon number 1, 2, 3, 4, 5, 6. Now you can try to remember a rule about left is up or right is down or whatever it is, but I think it's actually easier to just memorize the structure of the ring. You need to know the ring structure for glucose, fructose, and ribose. For glucose, I remember it as the alpha form of glucose is down, down, up, down, up. Down, down, up, down, up. See? It's easy. All right? Now, the name I was going to give you was actually relating to the ring itself. And the ring has an oxygen. There's a chemical known as pyran, or pyran, that has an oxygen like that. And so we refer to things that make six-membered rings with an oxygen in them as a pyranose. P-Y-R-A-N-O-S-E. Notice that the "-ose" at the end indicates it's a sugar. This is a pyranose. Tomorrow I'll show you the structure of fructose. And fructose can form a five-membered ring. We call that a furanose. F-U-R-A-N-O-S-E. We've reached the point of the lecture where I think we're going to sing a song. And there's a couple things I haven't covered in the song yet, but you'll hear those tomorrow. So please join me. It's a very easy song to sing. It's to the tune of Hark the Herald. Carbohydrates all should sing, glory to the Hayworth ring. Anomeric carbons hide when they're in a glycoside. Glucopyranose is there in the boat or in the chair. Alpha, beta, D, and L. Diastereomer hell. Alpha, beta, D, and L. Diastereomer hell. See you guys tomorrow.
I need to get my test on Friday.
Yeah, come by my office. I'll be happy to give it to you.
Like right now? Or like
If you want to, once I get the camera put away.
that would be, okay.
Okay?